eDNA stands for environmental DNA, and this is DNA that's been left behind animals or plants in the environment. What we're doing here is collecting eDNA from the sea, and that means that if there is a fish or a whale or a penguin or a seal that happens to have passed through that area that where we're filtering water, we might well be able to detect it after the animals passed out of the area.
The reason we're doing eDNA is because this technique allows you to detect species that you otherwise won't be able to observe, since they are really sneaky or too small to be observed from a boat or something like that. And the means to go in the water, like send divers to do recensement and all, it doesn't happen that often. And here from a ROV we can sample in half an hour and do an inventory of the entire surroundings, just like that with this technique.
So as an animal passes through this area where we're sampling, it leaves behind, it sheds skin, mucus, poo, whatever it is that it's left from its body, and we will hopefully collect it on our filter and get an estimate of species richness in this particular area. So we're using eDNA around Elephant Island because no one's ever sampled here before, and we're really hoping that we can get an accurate estimate of species richness in some of these areas. It's a novel technique, and what we hope to do is provide a real baseline of what species live here. And sometimes using eDNA can give us hits of species where it's very difficult to actually observe them. So we might find we find some cryptic species that haven't been recorded here before, and it also just means that if someone wants to come back to this area and sample in this way in the future, they can then compare those records from year to year, and it might tell us about different species that are here, say for instance invasive species or species that aren't found here. So we're really hoping that we can provide some baseline research for this area that can be useful for the future. When we left the Arctic Sunrise, we were heading towards our first transect or second transect of the day, and we were lucky enough to sight a humpback whale. And actually a minke whale as well. But the humpback whale was logging at the surface, so resting, and then at one point it fluked up and we were able to collect some photo ID shots of that whale, just of the fluke, which will be a really good way of individually identifying it as within all the whales that are in this area. There are catalogues of humpback whales around Antarctica, and what we will do is we will pass our photograph of our whale that we saw this morning and pass that on to other people working in the area so that they can try and identify where that whale was seen before, or maybe it might be seen in the future, and that'll all be because we managed to collect a really good photo ID shot. So what we're doing is we're just setting up so we can take a depth sample. So we have 2 Niskin bottles, which can then go right down to just above the seabed, and they're going to collect 10 litres of water each when we trip them with a messenger that we send down the line. And then we're going to retrieve them, pull them back up and filter the whole 20 litres of water through eDNA filter, and that should give us a sample right down at depth.